Hey there, deep divers. Ever just stop and wonder how your body actually pulls off?
Well, everything from a simple blink to, thinking about philosophy.
It really comes down to communication, doesn't it? But at a scale that's almost impossible to imagine.
Exactly. And today, we're diving into that hidden world, into the electrical language of animal life. It's fascinating stuff.
Absolutely fundamental.
We're going to unpack Chapter 4 of Animal Physiology, from genes to organisms. It's dense, yeah, but we'll pull out the key nuggets for you. Our mission, get you up to speed quickly on how cells, especially animal cells, talk.
And what's so striking is how universal this language is. You look across the animal kingdom from simple organisms to us, and you see these core mechanisms again and again.
Really conserved, you mean?
Incredibly conserved. It points to really ancient evolutionary roots for this whole electrical signaling system. It connects everything from the genes up to the whole organism's behavior.
Okay, so let's unpack this. Before we get to like consciousness or anything complex,
Yeah.
we need the absolute basics. Membrane potential, right, the starting point.
So every cell basically has this tiny electrical charge across its outer membrane, its skin, a separation of positive and negative ions.
Yeah, think of it like a microscopic battery.
Yeah.
This separation creates a potential, a voltage.
Yeah.
We call it polarization.
And that potential is there so the cell can, do work.
Exactly. It's the potential to do work, to send a signal, react to a stimulus, power processes. It's stored energy.
How does it get charged, this tiny battery?
Ah, well, that's clever. It's
Right.
down to an uneven distribution of ions. You've got lots of sodium ions outside the cell, lots of potassium ions, and big negatively charged proteins inside.
Okay.
And the cell membrane isn't equally open to all of them. It's differentially permeable. Some ions pass through easily, others not so much.
So that difference creates the charge imbalance.
Precisely. But then you have excitable cells, neurons, and muscle cells mainly. They've evolved to actively use this basic potential for really rapid signaling.
Like sending quick messages.
Exactly. I mean, get this. Even a single-celled paramecium, a protozoan, uses this. It bumps into something, zap. An electrical pulse travels across it, makes this little cilia reverse beat, and it backs away.
Wow. So this goes way back, evolutionarily.
Oh, absolutely. And we learned a huge amount about how it works from studying, believe it or not, squid. The loligo squid has these giant nerve axons, massive nerve fibers.
Big enough to stick electrodes in.
Literally. Pioneers like Hodgkin and Huxley did just that back in the day. It let them directly measure these electrical changes. Groundbreaking stuff.
So when this charge changes, there's specific language for it, right? You mentioned polarization.
Right. So polarization just means there's some potential. It's not zero. If the inside of the cell becomes less negative, say, it goes from zenith 70 millivolts towards zero, maybe to negative 30 millivolts. That's depolarization.
Getting closer to firing.
Kind of, yeah.
Yeah.
Then repolarization is when it returns back to that resting negative state.
And sometimes it overshoots.
It can, yeah. Sometimes it briefly becomes even more negative than resting, say, negative 80 millivolts. That's hyperpolarization, like a temporary dip.
Okay, got it. And how did scientists figure all this out with such precision? Squid axons helped, but...
Well, beyond just sticking electrodes in, techniques got really sophisticated. The voltage clamp technique was huge. It lets you hold the membrane voltage steady at a chosen level and measure the ion currents flowing across.
So you can see which ions are moving when.
Exactly. And then patch clamping came along, which is even more amazing. You can isolate and study the behavior of a single ion channel. Just one tiny protein gate.
Incredible resolution.
It is. And consistently, across all sorts of animals, the basic mechanisms involving these ion channels are remarkably similar.
Right.
It's all about ions moving through specific channels, these little protein gates.
And you mentioned different types of gates.
Yeah, 4 main kinds based on what opens them. Voltage-gated channels respond to changes in that membrane potential we talked about. Chemically gated or ligand-gated open when a specific molecule, like a neurotransmitter, binds to them.
Okay.
Then you have mechanically gated channels that open in response to physical deformation, like stretch or pressure. And thermally gated respond to temperature changes.
And this links back to genes.
Absolutely. Take voltage-gated sodium channels. They're crucial for fast electrical signals, and they're found almost exclusively in animals. The genes coding for them have evolved. Fast-moving predators or prey, they tend to have sodium channels that open super quickly.
Allowing for rapid responses.
Right. While more sluggish animals might have versions that open more slowly, it's a direct link between the gene, the protein's function, and the animal's whole way of life. Survival depends on it.
Okay, cell battery, check. Ion gates, Check. How does the signaling actually start? You mentioned a whisper.
Graded potentials.
Right, these are the first sort of local signals, graded potentials. They're changes in membrane potential that happen locally, and their size, their magnitude can vary.
So not all or nothing, like you hinted at earlier. Exactly. The stronger the initial trigger, maybe more neurotransmitter binding, opening more channels, the bigger the change in potential, the larger the graded potential. And the longer the trigger lasts, the longer the potential lasts.
How do they spread?
Just by passive current flow. Like electricity flowing through a wire, but in this case, it's ions flowing between the active area and the inactive areas right next to it.
But you called them whispers. Why only short distances?
Ah, because the signal fades fast. It's called decremental spread. The cell membrane isn't a perfect insulator, it's leaky. So, as the current spreads passively, some of it leaks out across the membrane.
Like sound waves dying out.
Good analogy, yeah.
Huh?
The further you get from the source, the weaker the signal. We can actually quantify this using Ohm's law and a concept called the length constant, lambda. It tells you how far the signal can travel before it decays significantly.
So they're not great for long-distance communication.
Not on their own, no. Though some neurons, particularly in arthropods called non-spiking neurons, actually rely entirely on these graded potentials for short-range communication. But evolution's found ways around this limitation for longer distances.
How? Better insulation.
That's one way you increase the membrane resistance. Or you can decrease the internal resistance by making the nerve fiber itself thicker, wider.
Bigger pipe, easier flow. Makes
Yeah.
sense. Which brings us to the long-distance calls.
Exactly. This is where it gets really exciting. Action potentials. These are the signals that can travel the entire length of a nerve fiber without losing strength.
The non-decreasing signals.
Right. Think of lighting a fuse on a firecracker.
Right.
As it starts, boom, it goes all the way down, self-propagating. Action potentials are like that. They're also all or none.
Meaning?
Meaning, if the initial stimulus, the initial depolarization, doesn't reach a critical level, the threshold potential, usually around -50 or -55 millivolts, nothing happens. No action potential.
Mm-hmm.
But if it does reach threshold,
It fires maximally.
it fires maximally every single time. Same size, same shape. This is really important because it filters out weak, insignificant background noise. Only meaningful stimuli trigger the signal.
So what happens during that boom, that dramatic change?
It's a rapid stereotype sequence. First, that slow depolarization reaches threshold, then bam, explosive depolarization. The membrane potential shoots way up, often into positive territory, like +30 or +40 millivolts.
Positive inside.
Yep, briefly. Then, just as quickly, it repolarizes, falls back down towards the resting potential. And often, like we said, there's that brief, after hyperpolarization, dipping even below resting before settling back.
And this is all down to those voltage-gated channels again.
Primarily, yes.
Yeah.
It's a beautiful, intricate dance between voltage-gated sodium channels and voltage-gated potassium channels.
Tell me about the dance.
Okay, so the sodium channel is the star of the rising phase. It actually has two gates, an activation gate and an inactivation gate. At rest, the activation gate is closed, blocking sodium entry. When the membrane hits threshold, that activation gate snaps open really fast. Sodium ions flood into the cell down their concentration and electrical gradients,
And that influx makes it even more positive inside.
which opens more voltage-gated sodium channels. It's a positive feedback cycle, very rapid, very explosive depolarization. That's the rising phase.
But it stops rising. Why?
Because of that second gate on the sodium channel, the inactivation gate. It's a bit slower, but soon after the channel opens, this inactivation gate swings shut, plugging the channel from the inside, stopping the sodium influx.
Okay, so sodium influx causes the rise. Sodium channel inactivation stops the rise. What causes the fall? The repolarization.
That's where the voltage-gated potassium channels come in. They also start opening around threshold, but they're slower than the sodium channels. So just as the sodium influx is stopping, these potassium channels are really getting going.
And potassium is concentrated inside.
Right, so when its channels open, potassium rushes out of the cell, taking its positive charge with it. This outward flow of positive charge makes the inside negative again. That's repolarization.
So sodium in for the rise, potassium out for the fall. Simple yet complex. What about that pump, the sodium-potassium pump? Does it play a role here?
Ah, good question. It's crucial, but indirectly for the action potential itself. The pump works constantly in the background, using energy to pump sodium out and potassium back in, maintaining those concentration gradients over the long term.
But not during the split-second firing.
No. The number of ions that actually move across the membrane during a single action potential is tiny. Really tiny. Like maybe 1 in every 100,000 potassium ions moves out. It's enough to change the voltage dramatically, but not enough to significantly alter the overall concentrations immediately. The pump cleans up later, basically.
Fascinating detail. Okay, so the signal is generated. How does it travel down the neuron? Neurons have different parts, right?
They do. You've got the input zone, usually the dendrites in the cell body, where signals are received. Then the axon hillock, right where the axon leaves the cell body, that's usually the trigger zone.
The decision point.
Yeah, often called the integrator or decision point because it has the lowest threshold, packed with those voltage-gated sodium channels. If the summed input depolarizes the hillock to threshold, an action potential fires. Then it travels down the axon, the conducting zone, sometimes very long distances. Finally, it reaches the axon terminals, the output zone, where it passes the signal to the next cell.
So how does it travel down the axon? Does it just flow like electricity?
Not quite like electricity in a wire because of that leakiness we talked about. In unmyelinated axons, the ones without that fatty insulation sheath, it uses contiguous conduction.
Meaning
The action potential in one patch of membrane triggers an action potential in the patch right next to it, which triggers the next patch, and so on. Like dominoes falling or that stadium wave analogy, each section actively regenerates the signal.
Okay, continuous regeneration. That sounds maybe a bit slow.
It is, relatively speaking, which is why many vertebrates and even some invertebrates evolved myelination.
The insulation.
Exactly. Specialized cells, Schwann cells in the peripheral nervous system, oligodendrocytes in the central nervous system, wrap layers of fatty myelin around the axon. But they leave small gaps.
The nodes of Ranvier.
The nodes of Ranvier, yes.
Mm.
These nodes are packed with voltage-gated sodium and potassium channels. The myelinated sections are insulated, so the current flows passively and quickly underneath them. And then it reaches the next node, where the membrane potential is still strong enough to trigger a full-blown action potential. So the signal effectively jumps from node to node. It's called saltatory conduction, from the Latin saltare to leap.
And that's much faster.
Oh, way faster.
Okay.
Up to 50 times faster than contiguous conduction. Plus, it saves energy because ions only cross the membrane at the nodes, so the pump has less cleanup work to do afterwards.
That's a brilliant adaptation. You mentioned invertebrates having myelin too. I thought it was a vertebrate thing.
Mostly, but surprisingly, myelin-like structures have been found in some earthworms, shrimp, even tiny copepods. It raises fascinating evolutionary questions. In insects, they don't have myelin, but they evolved a different strategy, a specialized nerve sheath, to help manage ion concentrations in their hemolymph, their sort of blood, especially if they eat plants high in potassium.
So insulation speeds things up. What about the fiber size you mentioned earlier?
Right, fiber diameter also matters hugely. Wider axon, less internal resistance, faster conduction. This led to the evolution of giant axons in animals needing really rapid responses.
Like the squid again.
The squid is the classic example used for jet propulsion escape, but also crayfish tail flips, fish Mothner neurons for startle responses. It's a common strategy for speed when myelin isn't an option or alongside it. A large myelinated axon can conduct signals at like 120 meters per second. Compare that to less than a meter per second for a thin, unmyelinated fiber. Huge difference.
Okay, signal generation, signal travel within a neuron.
Right.
But neurons need to talk to each other or to muscles, right?
Yeah.
A handover point, the synapse.
Exactly. The synapse is the critical communication junction between two cells. And there are two main types, electrical and chemical.
Electrical first. Seems simpler.
They are simpler. Basically, direct physical connections through gap junctions. Ions flow directly from one cell to the next. It's super fast, almost instantaneous. Great for synchronizing cells like in heart muscle or for really rapid escape reflexes.
But most synapses are chemical.
The vast majority in complex nervous systems, yes. Chemical synapses involve a tiny gap, the synaptic cleft, between the sending neuron, presynaptic, and the receiving cell, postsynaptic. The electrical signal can't jump that gap.
So it needs a messenger.
It needs a chemical messenger, a neurotransmitter. The arrival of the action potential at the presynaptic terminal triggers the release of this neurotransmitter, which diffuses across the cleft and binds to receptors on the postsynaptic side.
Slower than electrical, you'd think.
Slightly slower, yes, there's a synaptic delay. But chemical synapses offer huge advantages. One, they ensure the signal goes in one direction only. Two, they allow for much more complex signaling, excitation, inhibition, modulation, not just on.
Okay, walk me through that chemical handshake. What happens step by step at a typical fast synapse?
All right. Action potential arrives at the axon terminal. This opens voltage-gated calcium channels. Calcium ions, which are much more concentrated outside, rush into the terminal.
Calcium's the trigger.
Calcium is the key trigger. Its influx causes little sacs, synaptic vesicles filled with neurotransmitters, to fuse with the presynaptic membrane and release their contents into the cleft. That's exocytosis.
Okay, neurotransmitters out.
It diffuses across that tiny gap, it's really tiny, and binds to specific receptor proteins on the postsynaptic membrane. These receptors are often linked to chemically gated ion channels.
Ah, so the chemical unlocks the gate.
Exactly. Binding opens the channel. Now, depending on which ion channel opens, the effect can be excitatory or inhibitory.
Tell me about that difference. Excitatory first.
At an excitatory synapse, the neurotransmitter binding opens channels that let positive ions, mainly sodium, flow in. This causes a small local depolarization of the postsynaptic membrane. We call this an excitatory postsynaptic potential, or EPSP.
Bringing the cell closer to firing threshold.
Right. One EPSP usually isn't enough to trigger an action potential, but it pushes the cell in that direction.
And inhibitory.
At an inhibitory synapse, the neurotransmitter binding opens channels that either let potassium flow out or chloride ions flow in. Either way, it makes the inside of the postsynaptic cell more negative.
Pushing it away from threshold.
Exactly. This is called an inhibitory postsynaptic potential, or IPSP. It makes it harder for the neuron to fire an action potential. And crucially, the same neurotransmitter can be excitatory at one synapse and inhibitory at another, depending entirely on the type of receptor present on the postsynaptic cell.
Wow, flexibility. How is the signal turned off? Does the neurotransmitter just hang around?
No, that would be bad. It needs to be cleared quickly for precise signaling. It can happen in a few ways.
Yeah.
The neurotransmitter might just diffuse away, or it might be broken down by enzymes in the cleft, or it can be actively transported back into the presynaptic terminal or nearby glial cells for recycling.
Okay, that covers fast synapses. What about those slow ones you mentioned?
Right, slow synapses are super important for longer-term changes. Here, neurotransmitters like, say, serotonin don't directly open an ion channel. Instead, they bind to receptors that activate intracellular second messenger systems.
Like a cascade inside the cell.
Exactly. They trigger a chain reaction involving molecules like cyclic AMP, CMP, inside the postsynaptic neuron. This cascade can have all sorts of effects, modifying existing proteins, changing channel properties, even altering gene expression in the nucleus.
Altering gene expression, that sounds significant.
It is. This is thought to be fundamental for processes like learning and memory, where you need long-lasting changes in synaptic strength and neuronal function.
Let's ground this. A common example must be where nerves talk to muscles, the neuromuscular junction.
Perfect example. This is the synapse between a motor neuron and a skeletal muscle fiber. It's a specialized chemical synapse.
What's the neurotransmitter there?
It's acetylcholine, or SEC. When the motor neuron fires an action potential, it releases AC into the synaptic cleft at the neuromuscular junction.
And AC binds to receptors on the muscle.
Yes, on a specialized region of the muscle fiber membrane called the motor end plate. This binding opens chemically gated channels, causing a large depolarization called the end plate potential, or EPP.
You said large, larger than an EPSP.
Much larger, typically. The neuromuscular junction is designed for high fidelity. You've got multiple release sites, lots of AC released, a huge surface area with tons of receptors. So a single EPP is almost always large enough to depolarize the muscle fiber membrane to threshold and trigger an action potential in the muscle.
Which then causes contraction.
Which then causes contraction. And to make sure the muscle can relax quickly and respond to the next signal precisely, there's an enzyme in the cleft called acetylcholinesterase, ACGE, that rapidly breaks down HEE. It's a very efficient on-off switch.
Makes sense. You mentioned arthropods have more complex setups.
Yeah, it's interesting. Insect and crustacean muscles often receive both excitatory and inhibitory inputs from motor neurons, sometimes even inhibitory inputs onto the excitatory terminals themselves, allowing really fine control over muscle tension, quite different from our simpler vertebrate system.
Okay, so a single neuron is getting potentially thousands of these inputs, EPSPs and IPSPs, all the time. How on earth does it decide whether to fire its own action potential? It sounds like information overload.
It does, but the neuron is essentially a tiny calculator. It performs summation. All those incoming graded potentials, the EPSPs and IPSPs, spread towards the axon hillock, that trigger zone.
And they add up.
They add up, both in time and space. Temporal summation is when a single presynaptic neuron fires repeatedly in quick succession. The EPSPs, or IPSPs, arise so close together that they build on each other.
Like tapping faster and faster.
Exactly. Spatial summation is when multiple different presynaptic neurons fire at roughly the same time. Their individual EPSPs and IPSPs arrive at different spots on the postsynaptic neuron, but converge at the axon hillock and add together there.
So it's the grand total.
It's the grand total, yes. We call the combined potential from all these inputs the grand postsynaptic potential, or GPSP. If, at the axon hillock, the GPSP reaches threshold potential.
Bang! Action potential.
Action potential fires down the axon. If not, nothing happens. It's constantly integrating this barrage of excitatory and inhibitory signals.
Can you give a real-world example of this summing up?
Sure, the urination reflex is actually a great, if slightly mundane, example. As your bladder fills, stretch receptors send signals causing EPSPs in the motor neurons that control bladder contraction. A little bit full. Small EPSPs below threshold, more full. The stretch receptors fire more frequently, causing temporal summation of EPSPs. Eventually, they sum up enough to reach threshold, and you get the urge, the reflex extraction starts.
But we can control it, usually.
Right, because your brain can send down inhibitory signals, IPFPs, to those same motor neurons. These IPSPs counteract the EPSPs from the stretch receptors, that spatial summation of excitation and inhibition canceling each other out.
So you can hold it.
Exactly.
Oh.
Or, conversely, if you want to go and the bladder isn't quite full, your brain can send excitatory signals that spatially summate with the existing EPSPs to push the neuron over threshold voluntarily. It's a constant balancing act at the axon hillock.
That's actually a brilliant illustration of neural computation in action. So it's not just simple on-off signals. There's modulation, too.
Oh yes, lots of fine-tuning. We mentioned neuropeptides. These are often released alongside classic neurotransmitters, but act more slowly, often as neuromodulators.
Meaning they tweak the system?
Yeah, they don't necessarily cause a direct EPSP or IPSP, but they might change the postsynaptic cell's responsiveness to other inputs, maybe by altering receptor numbers or sensitivity. These effects can last much longer. Minutes, hours, even days. Many are also hormones, linking nervous and endocrine control.
More layers of control.
And there's even more localized control, presynaptic inhibition and presynaptic facilitation. This is where a third neuron synapses onto the axon terminal of the presynaptic neuron.
Influencing the influencer.
Exactly. It can release neurotransmitters that decrease inhibition or increase facilitation, the amount of neurotransmitter released by that terminal when an action potential arrives. It allows the nervous system to selectively filter or boost specific pathways, specific conversations between neurons without affecting the entire postsynaptic cell.
Super targeted.
Very targeted. And then there's the really cool idea of retrograde messengers. Sometimes the postsynaptic cell releases a chemical messenger that travels backwards across the synapse to affect the presynaptic terminal. Sort of like talking back, it adds another layer of feedback control.
This network sounds impossibly complex. How does it all get wired up?
Well, the fundamental patterns are actually quite simple, but they lead to immense complexity. You have convergence, where many presynaptic neurons contact a single postsynaptic neuron. This allows that neuron to integrate information from lots of different sources.
Pooling information.
Right. And then you have divergence, where a single presynaptic neuron branches out to contact many postsynaptic neurons. This allows one signal to be distributed widely, influencing multiple pathways simultaneously.
So convergence integrates, divergence broadcasts.
Exactly. These two patterns, repeated billions and billions of times, create the staggering computational power of nervous systems. Think about the human brain estimates are around 100 billion neurons, maybe 100 quadrillion synapses, all built from these basic signaling principles.
It's mind-boggling. But such a complex system must be, well, vulnerable, mustn't it, to things going wrong?
It absolutely is. And studying how things interfere with it, toxins, drugs, pollutants, even physical factors, has taught us a huge amount about how it normally works.
Like that pufferfish toxin.
Tetrodotoxin, TTX, yeah. It's a classic example. It physically blocks voltage-gated sodium channels. No sodium influx, no action potentials, instant paralysis.
But the pufferfish is okay.
It is because it's co-evolved. The toxin comes from bacteria it eats, and the fish has evolved a tiny mutation in its own sodium channels that makes them resistant to TTX. It's a beautiful example of an evolutionary arms race. Tarantula venom, ouabain from plants, many natural toxins target these fundamental electrical processes.
And drugs. You mentioned Prozac, cocaine.
Right. Many drugs work by altering synaptic transmission. Prozac increases serotonin levels by blocking its reuptake. Cocaine does the same for dopamine, leading to overstimulation of reward pathways and addiction.
Ooh.
Caffeine blocks adenosine receptors. Adenosine normally makes you drowsy, so blocking it perks you up. Ethyl alcohol, regular alcohol, enhances the effect of inhibitory GABA receptors, generally dampening neural activity.
Some toxins have really dramatic effects, right?
Oh yeah. Strychnine, a poison, blocks receptors for glycine, another major inhibitory neurotransmitter, especially in the spinal cord. Result: loss of inhibition, leading to unchecked muscle contractions, convulsions. Tetanus toxin from the bacteria works differently, but with similar results. It prevents the release of inhibitory neurotransmitters like GABA. Again, runaway excitation, muscle spasms, locked jaw.
Even environmental pollutants.
Yes, things like lead can interfere with neurotransmitter release, possibly by messing with calcium's role. It highlights how sensitive the system can be.
What about the neuromuscular junction specifically?
That's a common target too. Black widow spider venom causes a massive explosive release of Shi. Initially, it causes spasms, but then the muscle membrane gets stuck in a depolarized state, unable to repolarize and fire again. Depolarization block paralysis.
And Curer from poison darts.
Curer does the opposite. It blocks the ACE receptors on the muscle side. ACE is released, but it can't trigger the EPP, so no muscle contraction paralysis again, but via a different mechanism.
Yeah.
And then there are diseases like myasthenia gravis, an autoimmune condition where the body mistakenly attacks its own ACE receptors, leading to profound muscle weakness.
So chemicals are a big vulnerability. What about physical factors?
Definitely. Temperature is critical. Cell membranes need the right fluidity. Too cold, they get stiff, channels don't open and close properly. Too hot, they get too fluid, proteins can denature. Reaction rates also change.
And pressure, like deep sea animals.
High pressure is a huge challenge. It could affect protein folding, membrane fluidity, potentially squeezing channels or inhibiting pumps needed for ion gradients and action potentials. Deep sea animals have evolved amazing adaptations. Different membrane lipids, pressure-resistant proteins. Interestingly, marine mammals that dive deep but aren't adapted like deep sea fish might actually use these pressure effects. The pressure might naturally slow down nerve conduction and metabolism, helping conserve oxygen during a dive.
Wow, okay, what a journey. From the basic charge on a cell membrane through these fleeting graded potentials, the dramatic action potentials leaping down axons, the intricate chemical handshakes at synapses, all the way to this constant calculation and fine-tuning. It's just elegant.
It really is. This electrical and chemical language, built from relatively simple components like ion channels and pumps, allows for the incredible complexity of animal perception, thought, and behavior. It's the foundation of how animals interact with their world, shaped by millions of years of evolution.
From the gene level right up to the whole organism thriving in its niche. Amazing.
And, despite everything we've learned, everything we've mapped out about this electrical language, there are still so many unanswered questions, especially when we think about how these systems evolve in different ecological contexts, how they vary subtly between species, what other communication strategies are out there, what haven't we even thought to look for yet.
A humbling reminder that there's always more to discover. It definitely leaves you thinking. Well, thank you for joining us on this deep dive into the electrical language of animal life. We hope you found it as electrifying as we did.